Okay, so we're live here. All right. Well, thank you guys. Appreciate it. Let's sort of have you introduce yourselves and explain a little bit of your, background in what you're doing, and then we'll jump right into it. There's a lot of stuff we could go over here. So,
okay.
Yeah.
Okay. So I'll start off. So my name is Walt Golet. I'm an assistant professor at the University of Maine in the School of Marine Sciences. So for all of your listeners, the very, very far other end of the U.S., very far away from Hawaii. And myself and Sammi, Nadeau, who's also on this presentation with us, we work in the Pelagic Fisheries Lab. So the Pelagic Fisheries Lab was created by myself in 2011. And the focus of the lab is really to hone in on what I would call knowledge gaps or life history gaps. So these are things that we don't quite understand or don't understand enough of with regards to animals' biology, behavior, and so forth. And basically take those bits of information and then forward them on to the folks who do things like the stock assessments and set up management frameworks, all for the purposes of long-term sustainability, conservation, angling opportunities, and so forth. And one of the things that our lab really focuses on is industry collaborations. So I can't think of a single project that we currently have going on in the lab where we don't actively engage recreational or commercial fishermen and or both across all of these what we call highly migratory species. So the pelagic sharks that are managed internationally, all of our billfishes, inclusive of our marlins and our broad-billed swordfish, the tropical tunas and the temperate tunas as well. So our lab really focuses in on these industry collaborations. And we found that it's been a pretty successful model because it allows us to answer a lot of questions and gain access to a lot of samples, for example, that we just wouldn't have access to. So in addition to a research program that I run, I also teach a lot of classes at the University of Maine. Obviously, most of them are fisheries focused, fisheries biology, fisheries science, fisheries management, and so forth. And then the sort of the last hat that I wear is I try to stay really involved in fisheries management, both domestically and internationally. So I am on what's called NOAA's Highly Migratory Species Advisory Panel, which does a lot of domestic regulations with regards to marlins, tunas, sharks, and our billfishes. And then I chair the U.S. ICAT Advisory Committee. So for your listeners, if they're not aware, ICAT is one of 5 regional fishery management organizations. And these international organizations manage these highly migratory species in the different ocean basins. And in some cases, like the Pacific, because it's so large, you actually have a couple of these, what we call RFMOs. And ICAT is the one that does the management and assessment in the Atlantic. And for my part in that, I chair the U.S. ICAT Advisory Committee. And so in that capacity, we work with stakeholders. We work with the federal government. We work with states, nonprofits, ENGOs, NGOs, and so forth to basically come up with best practices, strategies for ensuring long-term sustainability of fisheries and protecting U.S. interests in those fisheries. So that's a quick rundown of myself, and I'll turn it over to Sammi, and she can give her introduction.
Oh, we can't hear you. You might. We gotta. It's the whatever your microphone is not working.
And if need be, I can step in for Sammi.
Well, she was muted. It was muted. Now unmute.
On your right side of the presenter. There it is. No.
I'm not sure why. Must be something with her. It could be something with her sound on her computer.
I'm more than happy to do introductions for Sammi.
Yeah, sure. And maybe she can.
Yeah,
she'll figure it out as we.
So no problem. So Sammi actually entered the lab, I believe, in 2018, and she came in as a graduate student. She was a master's student. She completed her master's a couple of years ago, working on the foraging ecology of Atlantic bluefin in the Gulf of Maine, documented some really important foraging changes that occurred. Her work is the precursor to the implementation of that into the management framework for some of the forage species that we have in the Gulf of Maine that bluefin are feeding on. And then once she finished her degree, she sort of graduated, if you will, and has been my lab manager ever since. And so she's the person that sort of keeps me on track and reminds me of all the things that I need to do and the deadlines that I need to make. And so she, in all seriousness, she basically manages the entirety of the lab. So we have somewhere between 9 and 12 different projects going on, personnel somewhere between 8 and 16 people during different parts of the year. And Sammi basically manages everything with regards to that, participates in the projects. For example, we'll probably touch on this later, but Sammi is part of the tagging initiative. She's tagged sailfish off of marlins off of the coast of Nantucket and off of Costa Rica, and has done bluefin tagging for me as well. So she's sort of a jack of all trades and keeps the circulation of the lab pumping.
Nice. Cool.
That's awesome.
Great. Well, we'll hopefully she will be able to get back and join us. I think she's just going to change her.
Change the mic out or something.
So it could
The other thing could be just like if she's doing headphones like I do, you have to change your microphone input on your computer. That's the other thing you have to do.
Yeah,
that might be it. But we'll I'm sure we'll get it worked out there somehow. But anyways, all right. Well, let's just jump into it, I guess. What might be kind of a cool thing to start off with is that the close kin work that you're doing, and you were going to you have a little bit of some stuff you can put up there as well.
Yeah, absolutely. I can share some great content with you. Just as a precursor to this concept, one of the challenges that is really ubiquitous across all fisheries, it doesn't matter if they are highly migratory fisheries or if they're ground fisheries, we all sort of are faced with the same challenges, and that is estimating or calculating accurate estimates of abundance. Right? In an ideal world, we would love to know how many fish there are in the ocean. But as I think of fisheries scientists claimed in the past, counting fish is like counting trees, except they're invisible, they move around, and they eat each other. So you're in a medium, right? And it's true, you're in a medium, and the expectation is that these scientists are going to be able to figure out what the biomass is. But they're trying to, it's a little bit like, detective work, because you're trying to solve the riddle, but you only have very limited pieces of information. And so oftentimes when an assessment comes out, it will have what we call some uncertainty associated with it. And then uncertainty is kind of baked into this because we don't know exactly how many fish there are. And so we estimate a number, and then that number has, some error around it or some uncertainty. So it's this number plus or minus this plus or minus that. And again, the more information that you can provide relevant to that species, probably the better you're going to be, the lower your uncertainty is going to be. But those sort of standard assessments can only take us so far in terms of reducing that uncertainty and understanding, sort of how many fish there are in the ocean. And so several years ago, scientists at CSIRO down in the Southern Ocean began to pilot or apply this concept of what we call close kin mark recapture. And they did it for southern bluefin tuna. And it worked. Actually, there's many of the authors of that are on here: Peter Grew, Mark Bravington, Campbell Davies, and so forth. And so this was a successful pilot in southern bluefin tuna. And the way that it works is, what you do is you sample adult fish. This is very similar to like a standard mark and recapture project where you would estimate, the number of fish in a lake by tagging some and then releasing them and then going back out and trying to recapture them. What you're doing is you're genetically identifying every single individual fish. So if you look on the left-hand side of the screen here, you'll see there's a bunch of adults. Well, just like us, every one of those adults has a unique genetic structure to them, right? That, you can tell you and I apart, not only because we look different or sound different or different heights, etc., but without knowing any of that, you could test your DNA and you could see those differences. We also see those differences in fish, and that's a good thing because that genetic marker, if you will, stays with the fish for the entirety of its life. So its fish's entire life, that sort of what we call a gene tag never leaves. its offspring also have those genetic markers, except those genetic markers are a reflection of their parents. And so what we can do is we can go out and we can sample adult fish, juvenile fish, and then we can sample on the right-hand side of this figure, you can see these little larvae. You can sample those larvae. And then what you can do is by estimating the ratio of genetically matched mature fish, so the fish that are on the left side of the screen, to the young of the year or the larval fish, you can use, again, more mathematics above my pay grade, but you can use this as a method to actually give you a population estimate. And if you can do this long enough and get enough quality data, the difference between the application of this and the application of some of our more standard stock assessments is that, in theory, this gives you an actual number. A lot of stock assessments will tell you what the biomass, or what we call the spawning biomass of the population is. Basically, it's a metric of weight. What is the weight of all of your adult spawners? Close kin is a little bit different in the sense that if you do this long enough and you get enough data, you can actually start to estimate the numbers of fish in the population. Again, you'll have a plus and a minus to that. You're never going to nail it exactly, but it appears as though it's a method by which your accuracy is much better and your uncertainty is much less. So the fact that this was piloted and successfully piloted for southern bluefin tuna was a sort of a game changer. And when those estimates first came out, those close kin estimates, they indeed did show that the biomass was higher than the biomass calculated in some of the historical stock assessments. So that created a whole nother host of problems, but those have been worked through. And so with the success of the application of that, folks at NOAA, folks with CSIRO, folks with Department of Fisheries and Oceans Canada, the U.S. academic labs like the Pelagic Fisheries Lab that we're in and so forth, all came together to try to apply this pilot to Western Atlantic bluefin tuna. And so larvae started to be collected. We started to collect the juveniles and the adults. Canada started to collect their adults. And we basically pooled all of these different samples together, sequenced each of those different tissues and larvae, and lo and behold, we found matches. And this has been headed up by Matt Loretta at the Southeast Fisheries Science Center. And I believe Matt said there was something like 55 or 56 matches already. So believe it or not, we've already been able to identify parents and their offspring. And through that, some of the initial estimates that have come out through close kin have shown something similar to what was demonstrated for southern bluefin tuna. And that is that our estimates from close kin, as opposed to the more standard assessments, are actually higher. And so we're really excited about this project, not only because the proof of concept worked, but we feel that, first of all, it'll probably reduce the uncertainty. It'll give us a little bit more confidence in those estimates. And secondly, what's really unique about this is it's an industry-driven project. And the way that it works is through collaboration between the industry, so the recreational and the commercial fishing groups and the commercial dealers, and the scientists. So the folks that are on the water, they're helping us to collect those samples. We're going out, we're getting those samples from them, we're sending them out, they get sequenced. Folks do the more advanced mathematics and modeling on this. And, this whole chain works together in order to create, these estimates. And it's like I said, it's a big collaboration. Here, we have larval cruises that are going on in the Gulf of Mexico. They're collecting the little baby bluefin. We have electronic tagging programs that are going on at the exact same time. And those tags on those fish are telling us where the bluefin are at certain times of the year, what stock they're from, what sex they are, if they're males or females. We also have the sampling programs on the right-hand side where it says fishery catches. These are areas where we're collecting the adults from. And again, you put all of that stuff together, and it allows us to create these estimates of abundance. And so it's really, really exciting. Here are some of those estimates. So you'll see there are these arrowheads here. All of these little colored squares are the larvae that were collected in different years. So red is 2018, blue is 2017, green is 2016, and so forth. And then all of the arrowheads, the corresponding arrowheads to the Northwest Atlantic, basically Gulf of Maine, Canadian Maritimes, are the matches. So that's where the parents were harvested. It could have been harvested lethally, or it might have been an adult that was captured, caught, sampled, and released. That's the beauty about the genetics program. We don't have to kill all the fish to sample them. You can let them go. All we need is a small thing. So what's really great about this program too is it doesn't just say, well, we're only going to get this for killed fish, which really restricts the sample size that you get. Now we can say, hey, anybody that's out there and you catch a bluefin, if you want to take just an extra 5 seconds and sample this fish from you, it's going to tell us a story. And you never know what that story is going to be. And so that's again, a really exciting part of this.
How would a recreational, how would an angler or somebody, if they wanted to participate or help out or anything, how do they get the information on that? What would they, what would they do? How would they find it all?
Yep, we can share it. That information is some of that information is right here. So we have a program called Genetics for Giants and Juvies, a little bit of a play on words, but it's a sampling program whereby folks can go out and they can sample these fish for us. Everything is free. We provide the industry with the sample kits, and inside each of those sample kits is going to be a pair of scissors to cut the finlet or cut part of the fin off. There's going to be individual files. There is a little paperwork. We do need to get some information from you. So I can't totally exclude people from paperwork. There is a little bit.
Yeah.
We do need to know things like, what was the date that it was caught, approximate size, location, and things like that. But it's really easy. You get in touch with us. We ship you a kit that has all the vials in it free of charge. We'll FedEx it to you. Once the vials are filled, you just let us know, text us, email us, say, hey, I've got a kit. No problem. We send you the FedEx slip and you can send those back to us free of charge. The one thing that I will say that's really important for this is for us to get these kits back. It's certainly wonderful to have all the enthusiasm and to send the kits out, but really where the rubber meets the road is when you send those back to us, because without that information coming back, we just don't have any way to tell what it is that you did. So if they're sitting on your wheelhouse or they're sitting in your kitchen table somewhere, they're not any value to us. So we always emphasize to people, just get in touch with us. We will help you get them back to us. And we've tried to make it as simple as possible. So, we have a lab page. It's just Pelagic Fisheries Lab. We're on Instagram. We're on Facebook. We have a UMaine web page as well. And you can find this information in a variety of different ways. As I said, you can see some of this information here. You can feel free to contact me directly, or you can go through some of those other avenues. And again, we've been very successful in the commercial fishery because the commercial fishery is a little bit more, I guess I would say, concentrated in the sense that we can work with not only the boats, but we can also work with the commercial dealers who have been sort of invaluable. They act like funnel points. But now, because it's non-lethal and we don't need the whole fish, we don't need the head or things like that. I mean, we can send kits anywhere. Florida, Georgia, North Carolina, Delaware, anywhere. And so we're really looking to ramp up the effort, especially on small bluefin tuna. And when I say small, I mean bluefin that are less than the commercial minimum size of 73 inches. So for all the boats that are in that, that angling category, and they do a lot of catch and release, or even they do some catch and retention, boy, those fish are super valuable right now. And we would love to get in touch with people and have them send those samples back to us.
Cool. And so currently that close-kin marker capture stuff is just with tunas, right? Is that the only thing you're doing?
There are other fisheries that have utilized it. I can't speak as an authority on all those other ones, but the ones that I know of for highly migratories, yes, those are the two tunas. So southern bluefin tuna is one, Western Atlantic bluefin tuna is the other. And when I say Western Atlantic, again, if folks that are listening are not familiar, the Atlantic bluefin population is managed as two different stocks. So we have an eastern stock and a western stock. And so again, historically there was a lot of information that suggested, we do have these two stocks. They're fairly separated, but we do know that they mix. Now I think we're in a space that not only do these fish mix in much higher proportions than we ever thought, but they mix differently by the size or age class that they are. There might be some mixing based on males and females. Now there's even talk that there's more than two stocks. And so a lot of folks are trying to put effort into using genetic markers and otolith chemistry and other means by which to identify if there are more than two stocks. Again, right now, bluefin are just managed based on whether the fish are to the east or to the west of the 45-degree west meridian. If they're to the west, then they're Western Atlantic, and if they're to the east, they're Eastern Atlantic. And again, a lot of that was based on historic knowledge that Western bluefin are presumed to spawn in and around the Gulf of Mexico, and Eastern bluefin are spawning in the Mediterranean. But now with the discovery of the Slope Sea off the east coast of the United States and the fact that bluefin are spawning out there, that's a whole new ball game. Now we have a completely new area that we need to explore and learn more about. And I think that's one of the most fascinating things about my interest in science is that just when you think you know it all, you don't know it all, and something else comes up, and it challenges your previous beliefs, and you have to be malleable and go with it and basically start to ask new questions or change your questions and say, well, I didn't think of that, or I didn't realize that, and now we have to kind of change our thinking.
Yeah,
Sounds like
yeah, that's really cool. In that chart that you have right there, right, the pairs, just so I understand if I'm reading it correctly. So all the dots here and everything, that's all juvenile or larvae.
Larvae. Yep, they're larvae.
So that was larvae, and then up here off the top, kind of up way in the northeast and all up in that zone, that is where the close kin were captured, or that is.
So the way that you read this is each of the squares, the colored squares, as you said, represent different years of larva collections.
Okay.
The arrowheads connect where a particular larva was sampled in the Gulf of Mexico, and then subsequently one of the adults that was a direct relation to that larva, in other words, it was a parent, was captured. So let's take this arrowhead here. This one, hopefully you can see my cursor, but the one that's on the southeast side of Newfoundland, sort of close to the Grand Banks. So that is an adult that was sampled, and that adult had a genetic match to a larva that was sampled down here in the Gulf of Mexico. And that's the way it works. That's what we call the short term is POPs, P-O-P, parent offspring pairs. And that's what you're looking for. You're looking for those direct descendants, if you will, of the parents. And again, I can't claim to know the mathematics enough. I am far from an expert in that. But there is a way to look at those ratios and to be able to calculate an actual abundance out of it. So yes, you're reading that figure correctly. The colored squares are the larva, and the ends of the arrowheads here are areas where an adult that was the parent of those offspring was captured and either killed or was captured and then released.
Yeah. No, that's really cool. So it kind of gives you an idea of where they're spawning, and then where they're full grown at, and what's going on there.
And this year we have plans. Working with NOAA, we were very excited about this. We have plans to do. Dave Richardson at the Northeast Center has plans to do larval surveys out in the Slope Sea. And if you don't mind, what I'll do is I'll scroll down to an area here that might give you a little bit better perspective. I'll just kind of show a quick maybe track. So let me just put this up real quick. So the Slope C, again, this was an area there was a published paper, I believe it was 2016 by Dave Richardson et al. And what they identified was larvae. And I'm not sure if you can see my cursor, but hopefully you can. They were identified off of North Carolina. So let's orient everybody. So here's North Carolina. And those larvae were found in an area that they deemed the Slope C, which sort of extends up off of George's, kind of comes out here. Forgive me, I might not have the demarcations on the eastern side exactly right, but it does kind of come out here and sort of encompasses this area. And the fact that there are larvae there and the larvae were so small was indicative that bluefin must have been using that area to spawn. It wasn't an advection or a drift of larvae from, say, the Gulf of Mexico. They were too small. And the timing was different. So now what's so exciting is you have this other group or this other possible stock, if you will, of fish. And there's going to be a concerted effort this year to sample larvae out there. So keep your fingers crossed, do a jig that Mother Nature is going to cooperate. And if that's the case, who knows what that's going to open up in terms of the relatedness of these pops. I mean, we may start to find additional parent offspring pairs, and that's going to be very exciting because that'll give us an idea of who's spawning where. So it's really exciting, and other things that come with close kin are you can start to see contributions of individual females through time. So you can get estimates of what we call fecundity by age. Fecundity is just the number of eggs per unit, like unit gram or something. And so it's basically tracking reproductive capability or reproductive output. And so you can if they live long enough, you can actually see the contributions of different individual fish. So it's very cool. And as I said before, now we can use genetics, at least in theory. This hasn't been fully vetted yet, but it's getting close. It looks like we can also use the close kin work to identify these differences in stocks. So east versus west, perhaps. And then even more than that, as I mentioned, we can identify the sex of the fish. So we can tell if it's a male or a female. So, from just a tiny, tiny piece of muscle tissue, or a tiny piece of a finlet, we get this just enormous amount of information. Yeah. And what's even better is it can all be used in the assessment for these animals. So I'm just really excited about it. I'm just really excited. It's a huge effort. It's definitely not anything that our lab is doing per se. We are participants in it. For certain, it's an international effort, and it's taken a lot of folks all the way from the industry on up to the people who are running the mathematical models to make this work. Without any link in that chain being complete, if there's a break anywhere in that chain, this project doesn't get off the ground. So it's just kind of a really neat success story, I think, in terms of industry collaboration and scientific collaboration.
And you're obviously able to tell age as well.
So here's another exciting thing. Let me share this. Sorry, I'm nerding out on all of this stuff.
It's awesome, man.
So age. So how do we tell the ages of a fish? I always joke with my students, right? You can't talk to them. You can't ask them. So traditionally, we've aged highly migratory species a couple of different ways, and we have to do it because of the logistics. So for tuna, most of the tunas, especially our tropical and temperate tunas, and a lot of our other what we call teleost fish or bony fishes, they're going to be aged with what are called otoliths. So inside the skull of the fish, there are 3 sets of otoliths. There's one on each side of the skull, and they're located in these little, this is a bluefin head, by the way. So what you're doing is you're looking at the top of a bluefin head with the top lopped off. So you're sort of looking down on the fish. Here's where the brain would be. And then in the back of the brain, you have these 2 little capsules. So these 2 little capsules here will house the otoliths. These are called, not to get too much into the terms, but these are called sagittal otoliths. And for tunas, they're the largest of the 3. And the cool part about these is, just like a tree stump, they collect, or they accrete rings. And so from the time the egg is fertilized till the time that the fish dies, material is accreted or accumulated in these otoliths. And the rate of deposition is different in summer and in winter. So when you have these temperate or seasonal migrations or fish that experience seasonality, that's really good, because then you get these more defined changes in the density of accretion. And just like going out in your yard and you cut down a tree and maybe sand it down or put a little varnish or shellac on it or put a little water on it, you use something to bring out the rings. That's exactly what we do with fishes in the lab. So these are examples. So these are the otoliths here. So what we do is we cut them. We cut a section. Well, first of all, we embed them in epoxy because they're really brittle. So we embed them in epoxy and we cut them in half. And when you cut them in half and lay them on your side or their side, you mount them on a microscope slide. And then when you look through the microscope, this is what you see. And so these are two bluefin that were rather old. The top one comes from the Department of Fisheries and Oceans. And I guess what I would do is if hopefully your readers can see this, or your viewers can see this, but if you look at the top part of this otolith, do you see how there's an area that's light and then dark and then light and then dark and then light? It's a little more prevalent over here. You can kind of see a little bit darker blue and a little bit lighter blue. They're like alternating lines.
Mm-hmm.
Those lines represent 6 months of life of the fish. So a dark one and a light one collectively are 1 year. So what we do is, we have protocols and things that are set up, but what we do is we go through and we count those lines, and that gives us the age. And so this is one of the older bluefin in the Northwest Atlantic that's been aged. It was 35 years old. And again, this is Alex Hank's group in Canada. And this was one from our lab that was 31 years old. So most of the pelagic fishes, the billfishes and the tunas, are what I would call moderately long-lived. They can obtain ages probably somewhere between 10 and 40 years old. Certainly some of the larger pelagic sharks, live a lot longer. But the tunas I put them kind of in the medium age range for most fishes. They're not like a Sebastes, a rockfish that lives for 100 years, and they're also, not something that lives only for a couple. But that's exactly what you do. This is exactly how you age. So bringing this back to your question, this is where it gets really cool. To do this process with otoliths, it's labor intensive. Our lab goes out every year and we collect somewhere between 1,500 and 2,000 of these bluefin heads. And actually here, in this picture, you can see it. So I think here there's like 100 of them laying on the lawn. They're big, they're heavy. And so we have to excise the tissues, clean them up, take them to the lab, prep them, and then, go through the aging process. Genetics offers potentially the ability to use what we call epigenetic clocks as a means to age fish. And it's complicated. It's very tissue specific. There are a lot of nuances there. But again, this work has been piloted for some of the tunas, and it looks like it works. So from a single piece, basically what you're doing is you're looking at this sort of DNA degradation. And so, just as we sit here and talk, right, we are degrading. Not to be morbid, but we are, even though we're alive, we're still sort of degrading. Well,
that's true.
You can use that information in the DNA as a way to tell age. And it looks as though it's pretty accurate. So what's amazing and in this as well is, like, let's say that you tag a fish and you let it go, or you catch a fish and you let it go. Once we vet all of these applications, right, of this technology, once they're all verified and everybody's on board, because you need consensus for this stuff, from a single fish, if you tag it and release it, or just release it, you should be able to get its stock ID, its age, its sex, and you'll have the genotyping for the close kin work. So you're going to get all of this information, again, out of this tiny piece of tissue. And it's amazing because it cuts down on costs, it cuts down on, labor time and all of these things. Now, I don't want to get too far ahead because we're not quite there yet. We're still working out some of the kinks. This isn't operational at this point, but the pilot projects for these look really, really good. So I think we're gonna have some good progress here in the next couple of years.
No, I mean that's fantastic. I mean, you're literally getting all the information. You can almost so much information without ever harvesting the fish. I mean, that just
Exactly.
changes work.
Yep. So you can get, so now, well, I shouldn't say now, I should say very soon, I think we'll be able to get the same information from a dead fish as a live fish. And how cool would it be to, you sample a fish, like, and it doesn't matter. Our ambitions are to go beyond bluefin. I mean, we want to do this with the other tropical tunas. We want to do this with billfish. We're talking with Peter at the Billfish Foundation about these initiatives. And we want to do this with other highly migratories. Tunas are just, bluefin is just one small piece of the pie. Actually, in terms of fisheries, bluefin's very small in the Atlantic and in the Pacific. But the idea is that, do this same thing. And I mean, you can imagine you're in a tournament or something, and you sample, you send your flags up, right? You're releasing all these marlins, and you send your flags up, and you come in with this information, and all of a sudden, 10 years later, you're in the same tournament. You send your flags up, you've sampled those fish, and all of a sudden, here's a blue marlin from that you tagged and released, or excuse me, that you sampled and released 10 years ago. Those are the types of things that we're hoping to be able to build, these sort of interactive databases where people can submit their samples, and then they can come back in and they can check on and see, did I get a match? Now, just because you submit something doesn't mean that we're always going to find a match. Truth be told, most of them won't have matches, and that's just probability. That's just numbers. But there are going to be some that are going to be matches, and that's kind of what sort of creates this. I don't know. For me, it kind of creates this really awe-inspiring or sort of very cool angle to this, because there's a lot we can give back with the information.
I have a quick question, not to
go ahead.
just based on what you obviously, as just from school and too, the more information you have is the best. But I would imagine too right now, the fact that you're not getting it, it's obviously you want the matches, but on some level, I would think if you're not getting a ton of matches, that might be kind of a good thing, because the stock is not so small that every time you get a thing, boom, But if you're not getting those hits, like, oh, actually, our stocks are doing good. We're getting proper. Our management plans working to some degree. Obviously, we want them to be better in the future, so for the future generations. But what we're doing is working to a degree that we're not. If we're getting hit every time, that's not good. That means either our larvas are not making, our spawners are super small, which is not what you want, obviously.
Yeah, exactly. Nope, you nailed it. No, you nailed it. You absolutely nailed it. If we, let's say we did the Bluefin project and we got 2,000 matches or 5,000 matches, and we only we only submitted 5,000 samples. That's an moment.
Yeah.
Or actually, I should say, do you have a bleep button on the show? It's an moment, right? I mean, that's like a no. So the fact that you don't have as many is good. Now again, that's going to scale. There are some nuances, there's some nuances to that, right? And you also have to deal with things like patchiness. For example, you might go through a patch of larvae that are they're very concentrated, not only in terms of abundance, but also they happen to be from 2 or 3 individuals or something like that, or 4 or 5 individuals. And then randomly you ended up sampling those couple adults somewhere. So there are nuances, but I would say, your general idea there is, if you're not finding thousands and thousands of matches, that's probably a good thing.
I feel like once you get the science at the upper end, like you're talking about, you would expect to get the many matches, but when you're at the lower end, if you're getting matches without all this other stuff, then it's like,
yep, exactly, yep.
That's cool. If it was, it would be really interesting if it goes into the billfish side of it as well. For a lot of our, we're still all trying to figure out what a lot of the billfish are doing, and
oh, absolutely,
it would be really cool. I mean, especially here in Kona, where the consensus is that this is a spawning area, that the fish are coming here to spawn. So seeing then that, okay, these, being able to then having larger fish that are in the 800 plus size that don't have to be killed, but they're larger fish, and then getting the DNA from them and saying, oh, actually, this is the same genetics of a lot of larvae that was found off the coast of the Big Island of Hawaii. And, maybe the fish was caught in the Marquesas or something like that. And it was very large. And we know that these big fish are coming in here to spawn and then continue moving on, which there is I'm sure there is some scientific data that's already been submitted and is saying that is why they're here. But that would be a pretty cool thing to see as well, because
Awesome.
yeah, that would be cool. I mean, and as from as an angler point too, you know, especially we release a lot of our fish, but to be like, you know, we caught a juvenile fish of this other one that was caught, you know, way down in Tahiti or somewhere like that. I mean, I think that's a really
and just thinking like it also just you could almost stack it up for your career, Chris. Like how many big ones you like caught and release. Like imagine you start catching like that big one we released. You also start catching her offspring like 10 years later. Like how
right? Cool that would be a really cool thing to be like, yeah, I let this one go. That might have been a grander, but we let it go, and now I've caught 7 or 8 fish that were its offspring or something, or other people have, or something, you know, and that's cool.
And some of the other things that this can do as well, which is really interesting. So one of the biggest challenges in fisheries is identifying age at maturity for some fish, not for all fish, but for some fish. It's definitely a challenge. That has been probably the biggest question for bluefin going back 5 decades now. The close kin is going to allow you, in my opinion, maybe others would argue not, but in my opinion, it's going to allow you to basically start to determine that information. Why? Well, because when you start getting the matches of the fish that are 3 or 6 or 5, eventually you're going to get to a point where you see parents that are spawning, parents that are spawning, parents that are spawning, and then all of a sudden at this cutoff, nobody's spawning, right? You're not getting any matches that are 3 years apart. So for example, if you have a larva and you have and you catch, we'll call it an adult, but let's just say a larger bluefin, and that larger bluefin is 4 years old, and you caught a larva that is genetically matched to it, there's no worrying about doing gonadal histology or any of the stuff that we used to do. You know, unless there's something anomalous about genetics that I'm not thinking of, you're going to know that is the parent of that larva. And so you'll know the age, and it starts to fill in those gaps. So like you said, you know, we can envision something similar for billfish, and we have a bunch of projects working for so you know, to get to the age question again, I guess what I would say is marlins have always been challenging to age. They use a variety of different structures. A lot of times they use the spines. They're the most accessible. They tend to have the best discrimination in those rings. So if you remember the otolith I showed you before, it has rings. The spines also have rings, and those rings correspond to, again, what we call annuli. But there are some caveats with using the spines, just like there are with tunas. They tend to have a mismatch between the number of rings and the actual number of years that the fish is alive. We've tried to use otoliths to age marlins and swordfish. The otoliths in marlins and swordfish are exceptionally small. They're exceptionally delicate, and they're just very difficult to work with. And so, you know, we hope to be able to, you know, we have a couple of projects right now where we're trying to validate the age of billfish, blue marlins, white marlins, and swordfish in the Atlantic. And second to that, we are trying to get away from the use of spines and use otoliths. And then again, maybe someday we can use genetics. I mean, we're not there yet. We're nowhere near that, but we have a project we're going to try to start very soon on swordfish epigenetic aging. So I guess what I would say for your listeners is that it's a really exciting time in the science field. We are just sort of poised, I think, to make a lot of really interesting discoveries. Not that we're there, and not that it's, you know, automatic, but I think that the collective through the science community is really at a stage where we can start to harness some of the power of this information.
It gives you so much hope for the future too. Like just working, like, you know, before, like you said, you're taking assessments, but now you get the easier and the better you can collect this information, the better the decisions you're going to make are going to be in the future.
Yeah.
It's just finally, it's just an awesome feeling, obviously.
Yeah.
Being good there.
And I think the other part that, again, not to beat this drum too much, but the fact that we can use methodologies and engage the industry is the key to this. I am one person. Sammi is one person. You are one person. There is no way that I can physically travel. And New England is tiny, right? New England is not big. Like our bluefin fishery, basically, for all intents and purposes, what is it? It goes in the U.S. It goes from about Carolina to Maine, to the Canadian border, right? Roughly, give or take, right? So in the grand scheme of things, that's not a big area. Still, there is no way that I could physically, or Sammi, or even a couple of us could possibly sample the number of fish that we need to. But with these types of applications where we can tell a boat, hey, get this for us, use this, now it opens up a whole new world. Now it's, we can collect fish here. We can collect fish 10,000 miles away over here, this side of the Atlantic, that side of the Pacific, this side of the Indian. I mean, it's kind of unlimited, and it gives you so much more access.
I would definitely beat that drum. I think that's the biggest thing. I mean, again, I'm very much on your side with that. I used to say that, like, when we were at schools, like, I think it's the biggest thing going forward. If you can get your recreation, all of those people on board, it gives you such a better view of what you're dealing with, like, so fast.
Yeah, it's a total game changer. So it's a total game changer. And not only individuals, but we also work with a lot of the tournaments. So we work with a lot of the billfish tournaments, for example. Again, very applied work there. I mean, this aging, I guess I would call it a validation study that we're doing with Alan Andrews, is utilizing a lot of tournament samples, things that were collected, for example, at, the White Marlin Open or the Mid-Atlantic, the Big Rock. Those are the tournaments. Now I'm only speaking for the Atlantic because that's where we are. But, these tournaments are obviously spread globally all across the world. And they just act as these great funnel points where we can be in one position. All the boats can come to us and give us access to. And a colleague of mine, retired colleague of mine from VIMS, John Graves, was a very prominent billfish researcher. He always used to say, marlins are a rare event species. That's not to say marlins are rare; they're not rare. But to a scientist, they are rare because we don't see them that much. We're only allowed to harvest 250 in the entire Atlantic for the U.S. So, and most people don't harvest that many, right? In our tournaments, we up our minimum sizes to avoid too many billfish landing. I mean, there's all sorts of things that we do to encourage catch and release, and only keep those individuals that we think are really in it for the competition. And so, for a scientist, they are rare event species. They're much harder to sample. So, tournaments are just another example of places where we can get samples. We finished a study. Actually, there's a paper in review now by one of my PhD students. We finished a study that utilized those three tournaments. We were asked by the Mid-Atlantic Council to do a project on the relevance of chub mackerel in the diets of highly migratories, and the only way we could do it for billfish was to go to those tournaments and sample those fish. Outside of that, we would have never been able to do it. It just would never have happened. So it's huge. I can't underestimate the participation of the industry, recreational and commercial industry, and in the case of bluefin, commercial dealers. But it's huge.
Yeah, and that was something we talked about when we just kind of had our call to kind of discuss what we were going to go over, was to kind of stress the importance of tournaments, of fishing tournaments, and how they are actually helpful. And I mean, the amount of samples that
I can give you a bunch of examples. I mean, I can give you a lot. So for the Mid-Atlantic, we have not Mid-Atlantic, but for the Atlantic, the U.S. East Coast and the Atlantic, like I said, those three tournaments have been instrumental for this, blue marlin, white marlin, aging study that we're working on. And by the way, that just to kind of give you an idea, I'll try to share this as well. So this is a project that we're working on. It's through Maine, but a colleague of ours, Alan Andrews, is the one that's really doing all the heavy lifting on this. And the neat part about this, I'll share it with you real quick, is the neat part is Alan actually uses bomb radiocarbon. So believe it or not, I don't know if I want to say this, that there's a benefit to detonating, nuclear weapons in the atmosphere, but I guess there is one here. And in the sense that when those detonations occurred, and hopefully I get this correct, Alan is the expert on this, but hopefully I get this correct, and this is taken directly from his work. There was an increase in this atmospheric carbon, and then there's this sort of steady decline that's occurring over time. And what you can, and this material is incorporated into things. It's incorporated into corals. It's incorporated, believe it or not, into fish otoliths, and we can use it as a means to confirm, for example, that those annuli, or those lines that I showed you before in the otoliths, right? Going the wrong direction here. So it's a way to confirm that these lines that you see actually represent a specific period of time. And so it's a really cool way to use something, that maybe wasn't the best thing to be doing as a means to confirm the ages in these fish. And so right now, Alan is working on this research, and he's got papers coming out with us on albacore. There's a blue marlin research project for the Atlantic that's underway. He already did this in the Pacific, and there's another one on white marlin and another one on swordfish. So we're doing the same thing on swordfish as well, with our colleagues at Fish Aging Services in addition. So it's just really exciting work. There's just so much stuff going on with regards to aging and billfishes, because we're really trying to refine it. We're trying to get the best information that we can out of billfish research, and aging is really hard. It's not a what we call strictly quantitative field. There's a lot of interpretation that goes into it, because you're using your eyes to sort of visually count, if you will, the rings on these structures, and they can be really hard, to do that. And so we can use this as a method to validate some of that work. And we did this in the Atlantic. Alan did this with us in the Atlantic, and he changed the age for yellowfin and big eye. Yellowfin, I think, went from 11 to 17 or 18. Big eye went from 15 to 18 or 19. So again, just when you think you know it all, you don't. And this new stuff comes on, and that work was incorporated into the tropical tunas working group and the assessment for those species. So it's just really exciting that you can see this work actually get used.
Yeah, that's pretty cool.
That's so awesome.
Yeah, that's definitely interesting about, though, that there's that you can from the nuclear they can see that in fish. I mean, it makes you wonder, like, we're eating fish, No,
you're good. You're healthy. You're good. Yeah.
Accumulating.
No, I don't want to alarm anybody. You're good. No atmospheric radiation fallout or anything like that. It's a very specific signature, and actually it will run out soon, because as you can see, the curve is starting to flatten out. So we're almost back down to previous levels. But it's a really neat way to get at some of the questions around aging for a variety of species. And Alan did this first, I believe, on a grander blue marlin in Hawaii.
Oh, wow.
Yep. And
I'll have to check out his stuff. His name, Alan Andrews?
Alan Andrews. Yep.
Okay.
Yep. Alan Andrews.
I'll have a look. He might be a great guy to
kind of. And he is a incredibly smart scientist, and he is sort of he's the authority. He's the authority that I work with. There are probably other authorities. I'm not singling him out, but he is definitely authority, and he's been great. He's done everything from HMS to demersal species to small pelagics and everything in between. So yep, Alan Andrews, and cool. He does a lot of this work, and I can share the paper with you if you want afterwards. But he did one of the grander blue marlins off of Hawaii, and I think his estimate was I shouldn't even say, it was well north of 20 years old. I don't want to say exactly because I don't remember.
That's kind of what I had always kind of heard or assumed it was 30 years or something. When
probably in that ballpark. Yep.
But that would be really interesting to have a discussion with him. So
he's great. He's great. He would be really good. And cool. he could do the research justice. I am sort of just touching the frosting on the top of the cake. enough to not get myself in trouble by saying the wrong thing. By saying the wrong thing.
Yeah. Oh, that's cool. Cool. Right on. Well, we'll kind of keep on moving along here.
Sure.
We went through some when we kind of spoke. You showed me some of the migration charts.
Sure.
And
Sure.
I know that some people may find that really interesting,
Yeah,
particularly
absolutely.
yellowfin ones and stuff like that.
Sure.
So yeah, so you guys are involved in tagging programs with yellowfin as well.
So we have tagging efforts for big eye, yellowfin. I think we're going to get into the swordfish game this year. We have the white marlin, the Skilly project with the Billfish Foundation, which is very exciting.
Yeah.
Yep. Bluefin tagging, obviously. We're doing some shark tagging now. Most of that is local. The shark tagging is local in the Gulf of Maine. But yeah, we've been branching out and trying to tag a lot of different species as of late. And let me see if I can bring up. I'll bring up a few of these different ones for you. And we can start with, if you want to start with yellowfin, we can start with some yellowfin ones.
Yeah, whatever.
Okay. So let me preface this by saying, and I'll just strictly talk about yellowfin in the Atlantic. Okay, this does not include yellowfin in the other ocean basins. It's yellowfin in the Atlantic, and one of I guess the most interesting aspects of yellowfin in the Atlantic, and this data comes from Dirk Snodgrass from the Southeast Fisheries Science Center, and ultimately it comes from ICAT's database, is that yellowfin in the Atlantic, I believe, are the only yellowfin around the globe that undertake such extensive cross-basin migrations. And so what you're looking at here are what we call conventionally tagged, so streamer tags, spaghetti tags, people often refer to these things, yellowfin tuna. This one up top here might be a bluefin. It went to the Bay of Biscay. That makes me skeptical. But the rest of them here are yellowfin. And so these are yellowfin that were tagged off the east coast of the United States. And a proportion of those were subsequently recaptured in the Gulf of Guinea. Now, here's some interesting things about the Atlantic. We know that yellowfin, as I said, go from one side of the basin to the other. This seems a little bit unique because in the Pacific and in the Indian Ocean, yellowfin certainly move, but we don't see, at least if I'm remembering the literature correctly, we don't see the same magnitude of what I would consider long distance migrations in a linear direction. So east coast of the US to West Africa, right? The Gulf of Guinea is this area down here where my cursor is. The Gulf of Guinea is considered to be the largest tropical tuna spawning ground in the Atlantic, as of right now anyway. But yellowfin spawn all throughout the Atlantic Ocean. They spawn off of Venezuela, they spawn in the Gulf of Mexico, they spawn in the Caribbean, and so forth. And yellowfin we know even less about. We manage them as one stock, but it's possible that there's multiple stocks. We just don't know. And so tagging is one of the ways that you can investigate, where are these fish going? Do they have residency? Do they have what we call fidelity? In other words, they're going back to certain places for certain reasons, whether it's feeding or spawning. And this was an effort by the US and other countries to tag yellowfin. And the conventional tags show that these animals do go from the east coast of the US all the way over here to the Gulf of Guinea. Now, why is that important? Just like with bluefin, in the Atlantic anyway, again, I'm not speaking for the other ocean basins, but in the Atlantic, this is a classic case of what happens on one side impacts the other side. So with regards to bluefin, we are the benefactor of a large Eastern subsidy. In certain years, upwards of 70% of our bluefin in the West Atlantic actually have a Mediterranean or an Eastern origin. So huge subsidy. And when you look at a figure like this, it makes you wonder the same thing. There's a huge spawning ground off the Gulf of Guinea. Like I said, it's not the only one, but it is a very large one. It's heavily fished by purse seine fleets and longline fleets throughout the Atlantic. And so there's this connectivity. Now, these were older tags. These were primarily from, I believe, the 1980s and 90s, maybe early 2000s. And since then, ICAT initiated another conventional tagging program, and we headed that part, and we too had fish that were tagged in the east, tagged in the Western Atlantic. Sorry, from the ICAT perspective, we call the east coast of the United States the Western Atlantic, but those fish were tagged off the east coast of the U.S., and we had 2 of them that were captured here in the.
You might have to. Sorry, you may have to share your screen.
I'm sorry. Oh shoot.
There.
Oh my gosh. Oh my gosh. Oh my gosh.
I thought that was just me.
Honestly, I thought it was
my computer's melting again.
There's no way.
But we can hear you, Samantha, so that's good. We can hear
That's
you.
good. Can you see me?
I am so
Yeah, I
sorry.
can see you too.
Yeah, you see.
So
okay, great.
let me start this again. I'm so sorry. So
Great.
quick recap. So what you're looking at are the conventional mark or tag, release, and subsequent recaptures of yellowfin tuna tagged on the east coast of the U.S. And you can see that there's this sort of highway that goes from Massachusetts, New York, Delaware, Maryland, Virginia, North Carolina to the Gulf of Guinea, which is located here off of West Africa. And again, that's one of the major spawning grounds. So there seems to be this connectivity with yellowfin between the East and the West Atlantic. We obviously, as many of you know, there's a very large yellowfin fishery in the Gulf of Mexico. The interesting part about that, it doesn't seem to mix with the other areas of the Atlantic. We see no movement of Gulf of Mexico yellowfin into the Atlantic proper, or vice versa. And we have tagging data. Now, this tagging data is a collection of our efforts. It's a collection of the NOAA efforts. It's a collection of a variety of different labs that have all put their data together. And one of my PhD students, Joe Della Russo, is working on this for his dissertation. And these are all electronic tags that were put on yellowfin. And you can see, for the most part, except for that one at the end there, that sort of blue line, most of these fish either are in the Atlantic proper, or they're in the Gulf of Mexico. Or even in the Caribbean. There's not a whole lot of exchange. There's a few caveats with the data. There are a few caveats with the data. And the biggest one that I'll mention is that compared to this graph, time at liberty for these electronically tagged yellowfin is much shorter. What the information suggests, at least this is one of the questions or one of the hypotheses we have, is that yellowfin may come over to the east coast of the U.S. when they're, as they're smaller, make their way over here. They may spend a year or two in the Western Atlantic. Maybe they do spawn on this side. We don't know. But after a period of time, and sometimes that's a year or two or more, they do make that journey back to some of them make that journey back to the Gulf of Guinea. And so one of the initiatives that we want to focus on now is longer term tags for yellowfin. A lot of like the pop-up, what we call the pop-up satellite tags that are used for many fishes. They're good. They're great tags, but the problem is they only stay on for a year. So if your question is directly related to something that you can answer within a year, great. They're a perfect platform. But if you want to answer something that's maybe multi-year, 3, 4, 5 years, then you use an electronic tag, but you use one that's a little bit different. And so our feeling is that a lot of these yellowfin are probably going to make that transatlantic journey. It's just they haven't been given enough time. A lot of the yellowfin that you see here are on the smaller side. So it's very possible that, the bigger they get, the more likely they are to maybe make that journey. Again, these are just kind of, hypotheses. But we don't have, on the short-term tags, we don't have any of them that are making that journey across the Atlantic. In fact, none of them even go to the mid-Atlantic ridge, let alone the west coast of Africa. And those Gulf of Mexico fish, Brett Falterman has a really great data set from the Gulf of Mexico. He has a ton of electronic tags. They tell the same story. Very, very limited, if any, mixing of those yellowfin outside the Gulf of Mexico. They seem to stay there. They have a lot of permanent.
We've been saying that for years. I mean, I'm from that's what I feel like all of our tunas are resident tunas.
Yeah.
Just
Yeah, a lot of talk about the tunas there being residents.
That's interesting. I wonder what the reasoning is.
Yeah, I don't know. I mean, there's one you see that goes out of the Gulf of Mexico there, but you're always going to have that. I mean, in ecology, you always have the anomalies. You always have the I call them the the rogue teenagers that just want to get out of town, right? They just want to leave. That
Yeah.
always happens. That always happens. But yellowfin. So there are some concerted efforts to tag yellowfin. We're not the only lab that does this. As I mentioned, Brett has a great data set on this in the Gulf of Mexico, and there are other researchers that are doing this tagging. ICAT did a fair amount of this electronic tagging as well a few years ago. So we're really trying to understand more about tropical tunas, because we honestly really don't know much about them at all. I mean, we know far less about them than we do bluefin, and I can't stand here and say that we know a ton about bluefin. We know a lot. Certainly, we know a lot more in the last 15 years than we have historically. But the tropical tunas definitely are lacking in understanding of life history, for sure.
Yeah. So the data kind of ends once they get over to Africa there. That would be sort of the longevity of the tags. And
see, in this figure, what happened is you had a fish that was captured, it was tagged, and then it was released, and then it swam. Let's just pick this one right here. It swam. I think that's Gabon. It swam to here off the coast of Gabon, and then it was recaptured. It probably was recaptured in a purse seine net, and they reported it. They reported the tag.
Sure.
So that fish is dead. It's that tag. Okay. That tag is very simple. It's just like the billfish tags. It does all it does. It tells you point A, point B. That's it. And so every arrowhead you see here is what I would consider to be a termination point, unless the fish was released. But most of these fish down here are not going to be released.
No, especially off there, they ain't getting released.
Huge purse seine effort off the west coast of Africa. It's massive. Unfortunately. Yeah, it's massive. So there's increased efforts to try to do some of this longer term yellowfin, tuna tagging.
Yeah, because that's kind of what I always see. Like even with the billfish stuff, it's like all the migrations for the most part, it's just a straight line. Not a straight line, but it's just a beginning and an end point. And I always wonder in my head, like, what are these fish doing for their life, their whole life? Like, are they just migrating to one area and then go there, and then they're staying there because the abundance of food and the situation's good, or they go there, spawn, do their thing, turn, and then they're going to another place and they're doing a cycle. That's what I'm always curious of, like, what is the life of, I mean, I'm a billfish guy, so I'm always thinking about billfish, but I'm always kind of thinking like, what are they doing? All the ones they tag off here seem to just go straight to the Marquesas or down south in that way. And either get captured or the tag pops off or whatever.
Yeah, I mean, we tested a couple of pop-up tags for Wildlife Computers. This is a long time ago. This was in Bermuda. Actually, I think we tested 3 of them. They were a new prototype. We had one white marlin. These were only 30-day tags. They weren't programmed to be on the fish very long. It was a short-term tag on purpose. But we tagged 3 white marlin. One of them went in 30 days, was up on the Grand Banks, so went north. 2 days later, another fish that was tagged in the same area actually went down by St. Kitts in the Caribbean, and then another one went west over towards North Carolina. So, like you said, it's amazing. It's amazing to think about, like, what are they doing? Why did the 2 white marlin that were tagged within miles of each other, only a day or 2 apart, one went almost due south, and the other one almost went, I'm exaggerating a little bit on the compass, but almost went, due north, right? Like they did this completely opposite thing.
Yeah. That is strange. It's like there's less consistency.
Yeah. I mean, here's a 10-month tag, or actually I think it's a 12-month tag, 10 or 12-month tag. 10 months, 10-month tag on a yellowfin that we put out a few years ago. And again, this guy just sort of left the continental shelf, went out to the seamounts, kind of milled around out there and came right back to where you see that red dot, and then the tag popped off. And then other fish are going across the entirety of the Atlantic. Why is that? Why is that? What's the motive? I mean, some of these questions we'll probably never know the answers to, at least in my lifetime, but it's like, what's the motivator behind that? Yeah. Why do you tag two fish in the exact same school and they do totally opposite things? Right. We have that with bluefin. They go complete. We have one fish, we've got bluefin tags where you tag two fish on the exact same day, presumably out of the same school, because we tag them in the same location, only 15 or 30 minutes apart. And you have one that goes to West Africa and you have another one that goes to the Gulf of Mexico. It's like, how in the world do they know? Like, how do they know that? That they got to go back and not to get off on too much of a tangent, but one of the things that I always impress upon my students, and it's so fascinating with tagging, how did they do it? I mean, if you're not to take anything away from birds, because birds have magnificent migrations, but they can cheat a little bit, right? They can cheat a little bit. They have landmarks, they have mountains and trees, and I think there's even some data now that suggests birds might even use roadways and stuff to navigate. Obviously, that historically wasn't what they used, but there are, terrestrial landmarks, I guess I would say.
Yeah.
But think about this for a second. You release a marlin, right? You release a marlin out of the Hudson Canyon, which is, off of the east coast of the U.S. And the next thing you know, that blue marlin is down in the Caribbean, or it's off of West Africa somewhere. And these fish do this with regularity. Like, they can make these round trips and they can go back to the same spots. They are in a medium where at best you might be able to see 100 feet. I mean, I did some scuba diving in Hawaii. I think the visibility the day we went out was like 100 feet, maybe 110 feet or something. It was a really good day. But, off of many of these shelf systems, the visibility is terrible, right? I mean, and this fish, think about this. This fish is seen in front of it, maybe 30, 40 feet, right? And yet it's able to navigate the ocean and go to presumably a place it's probably already been, and then come back again. And it's like, how in the, I lose my car at the store, right? I go get my groceries. I can't remember where I parked my car. This fish is traveling linearly, 10,000 miles and doing it with amazing precision.
Yeah. And finding food along the way too, and finding like rich areas, rich in food. That's the thing that trips me out with marlin, at least, is I'll go to a ledge that the current's good on and the bait's not really there yet, but the current's good there and it's going to get good. And I'll show up there. And as soon as I show up, there's a blue marlin. Like, how did it know to go and see that's the current and everything's good in that area? I'm going to go hang out here and I know I'm going to get food here soon. Like, I don't understand it either.
Yeah, it's pretty amazing. These are just some more yellowfin tracks off the northeast of the United States that are kind of illustrating, just sort of these 1 year movements or 1 year or less movements as well. And we have, again, this is a collective effort. This isn't just our lab, but I'm not sure, some folks might be, interested in some of these ones that we have for bluefin that I can show that are pretty interesting. Same thing, gets to the topics of what we just talked about. I can go into a little bit more detail.
You should scroll down too and talk, since we're talking about how do they know all this stuff, scroll down and show them the brain.
Oh yeah. So I do a lot of fishing seminars. I do a lot of them on the East Coast, and I always end with this. This is my ending slide, because I am not as seasoned a fisherman as some, but I've spent a lot of time on the water, grew up in a fishing family and so forth. And one of the things that I always, if I can find it here, that I always emphasize is, there's all this care that's taken to rig the baits and hide the hooks. And, for bluefin, they're always wiping down the lines and doing this. And this is what I end with. I always tell everybody, well, for all your efforts to try to fool the fish, this is what you're up against. And so this is the brain of a 600-pound giant. That's it. So this is the CPU, for example, that you're trying to fool with your your different baits and your different lures And this is also the same brain, mind you, that can do those migratory feats that I just described, where they can go across the ocean, and it seems like they know where they are. How is that possible? So this is what Sammy was referring to. That's the size of a giant bluefin brain. Marlins aren't any much bigger, by the way. They're about the same size.
Hasn't popped up on mine yet. I don't know if you guys see it.
Yeah, no, I don't see that. But...
Oh, I got to hold on. Sorry, God darn it! I have to share the individual screen. I'm so sorry.
I don't know. It didn't pop up from mine.
Hold on. Here we go.
No, you're good.
There we go. How about that?
It'll take a second. There it is.
There it is. So to reiterate again, I'm sorry about that. I had to change PowerPoint.
You're real smart now.
Yeah, exactly, right? I mean, so put your brain next to this one, and let's compare computing power.
Chris, you need to get a new job, bro.
So again, just to reiterate, this is the brain of a giant, and they can perform these amazing migrations, and at the same time, we go through all the trouble of presenting the baits properly and things like that. But I'm making light of it. I'm joking a little bit and making light of it. But for anybody that's spent time on the water, fish are not they're not stupid when it comes to hunting. They definitely can key in on certain baits, and maybe those baits are just a little bit further. Like, why do you have that stinger at the end for trolling for bluefin or tunas, right? Well, instinctually they know they're gonna go after that back one. Not all the time, but they go to that back one, right? That's the one. Why? Well, in theory, it's because it's the weakest. It's trailing. It's the one that's behind the pack.
Yeah.
So there is method to the madness here. I mean, that brain is doing stuff. So I'm not trying to make light of all of the things that are done in the fishery in terms of rigging the baits, because it does matter. It actually does matter. My point is, what's amazing is this brain can make it matter. That's what I find amazing.
Yeah. Oh, that's crazy. I'm glad you shared that. I knew they had small brains, but I did not realize it was that small.
Yeah, that's a 600-pound giant right there. So about the size of a dime.
Dude.
Wow. And yeah, the fish brain is, I think it's probably right around the same size, I would think.
Yeah, I think it's small.
Those things have been making me look stupid forever. So
don't even talk to me about yesterday, Chris. I don't want to talk about it.
Oh, well, we will.
But I can change slides again if you like, and I can talk a little bit about some of the bluefins, the bluefin migrations. I can share some swordfish migrations with you as well.
Yeah, I think they would
share some swordfish migrations with you.
Yeah,
so for bluefin, we have this idea with bluefin that they spawn historically that they spawned in one of two places. It was either the Gulf of Mexico, and I call it region because it's not just the Gulf of Mexico, and then the other was the Eastern Atlantic in the Mediterranean Sea. But now, as I said, with the discovery of the slope C and so forth, and the data from electronic tags, now we're left with these additional questions. And one of those additional questions is take a look at this fish. This was a big fish that was tagged in the Gulf of St. Lawrence, and it spent 10 months and it never visited a spawning ground. Well, let me qualify that. It never visited a known spawning ground in terms of the Gulf of Mexico, or the Mediterranean. Could it be a slope C spawner? Yeah, it could. It could definitely be a slope C spawner because these regions out here are part of that, that slope C area. So this might be an example of one of these fish that's actually utilizing this newly discovered or newer discovered spawning ground. There are other possibilities. It could be a fish that skips spawning. Very doubtful, but it's a possibility. Go ahead.
Where all of these fish are together, these are a bunch of different tagged fish.
One single. This is a single individual. Yep.
And so the colors are different months then.
Yep. Exactly.
Okay, wow. So he just kind of cruised around, stayed in that one little area, went up, went over.
Yep. And then the tag popped off right here in the middle of the Gulf of Maine in June.
Okay. Wow.
Yep. Pretty crazy, right?
Yeah.
And then you have this fish tagged in the Gulf of Maine. It was tagged right here where the green is in September. And again, very likely could be a slope sea spawner because it's utilizing that Carolina shelf all the way out here and this eastern edge of Georges. And then the purple is where the tag popped off, which is right there. So within a few miles of where it was tagged the previous year. So that fish basically came back to almost the exact same spot. So it was tagged here in green in September. It did its 10-month journey, went down to Carolina. Here it's down in Carolina, January, February, March. April, it starts to come up and then spends May, and then June starts to come into the Gulf of Maine, and the tag pops off right there. So probably, again, one of these slope sea spawners. At the same time, we have another fish tagged in the Gulf of St. Lawrence, and look where this one went. This one tagged in almost the exact same area as this one.
You might have to click on
Now
that
when,
slide.
whoops,
Yeah,
sorry, what?
Doing it again.
Sorry, is the slide up?
We're at slide 1 right now.
Slide 1, okay.
Or just box 1 or whatever on the left.
Okay, let me see if it goes to the next one. Or I can just force its hand and do it like this. And then you have...
There you go.
Yeah, here we go. We're at 3.
Okay,
Shows
perfect.
across the Atlantic.
Yeah, and then you have another fish, another bluefin tagged in the same area as that one that visited, that didn't go anywhere. And then the one, North Carolina, and then this one goes through the Straits of Gibraltar and ends up in the Mediterranean.
Okay.
Tag same time period. And then here's another one. Did it flip to the 4th slide or no?
Not yet. Sometimes it takes a second, but
Okay. I'll give
yet.
it a second. And if not, I'll change it manually.
Yeah, you may have to.
Okay, that's no problem. I can do it real quick. Here we go. Now that I'm familiar with the controls.
Yeah. Oh, wow. This one covers some ground.
Yep, exactly. So this one, same thing, tagged in the Gulf of St. Lawrence here in October. Went out around Cape Breton, came down, went through the Straits, presumably, went to the western Gulf of Mexico over here by Texas and Mexico, and then started that return journey. And what's crazy is, check this out. So it got from here. So this is May. So it started to head out of the Gulf of Mexico around the middle of May. And then this is May, and then this is June, and this thing is beelining it for the Gulf of St. Lawrence. And then the tag came off right there.
Dang.
Over here. So again, came off fairly close to, where it was
started.
tagged.
Yeah.
Yep. Fairly close to where it was tagged. And there's lots of examples. We're not the only one. There are other labs that have examples of this. It's just, again, like I said, it's really fascinating. And then we have, again, with some of our colleagues, if you're interested, I can show you some swordfish tracks.
Yeah, for sure.
From some of our colleagues. Got them here somewhere. So let me share this one. Sorry, just got to go through the proper steps this time, so I'm not talking about something you can't see. So hopefully you can see this one. Can you see that one?
Yep. It says swordfish.
Yep.
Collective effort.
So, yep. So this is again, a collective effort by a lot of different folks. And you can see just, some of these are shorter term tags. They didn't stay on the fish as long as we'd like, but some of them make this really, really long migration up to the Grand Banks, south of the Grand Banks, southeast of the Grand Banks. And, this one comes right back. This one here comes back into the Caribbean.
Wow, so went all the way up there.
Yes.
Turned around and then came back.
Exactly. Yep.
Wow.
Absolutely.
Dude,
Yep. Yep. And the reason, to get into the biology a little bit, the reason why these animals are doing these impressive migrations is because, the foraging is much better in the temperate latitudes than it is in the subtropical zones. So even though it's a large energetic cost for these animals to go north, right? They incur a cost in migration energetics. Yeah. Being there allows them to accumulate much more of lipids or much more fats than they probably would ordinarily be able to do. So it offsets itself by a lot. I don't know the weight gain on swordfish or marlins. We haven't studied that, but I can tell you on something like a bluefin, I mean, these animals can add, 150 or 200 pounds in a season up in the Northeast. So yeah, so they can add considerable, and swordfish add fat, marlins add fat as well.
And so the swordfish data too that you're getting is they're not crossing the Atlantic at all, huh? They're staying.
So far they're not. And this by the way, sorry, before I go any further, can you see that I switched slides or no?
Not yet.
Okay.
No.
We're gonna do this manually because I want to make sure I don't start talking. So to answer your question about whether we see these swordfish going across the Atlantic, so far no, we haven't. There are 3 stocks of swordfish. There's a Mediterranean, a North, and a South Atlantic. And so far, most of the tagging effort on both sides has indicated that these fish tend to stay on their respective sides, which, maybe that has implications for management. A lot more tagging has to be done on swordfish. But John Nielsen's work in Canada many years ago, his fish basically did a pattern very similar to this, where they were they would leave the Straits of Florida or East Coast of Florida, Caribbean, and they would go up towards the Grand Banks and come back, and they didn't cross the line. And I think a fair amount of the tagging data for swordfish on the Eastern Atlantic have done the same thing.
Cool.
They stay on that side.
That's interesting.
Wow. That's
I didn't know that.
Keep going. I have some more to show you. Here's another one. I think this is a separate one. Yeah, this is a different one. This is the one that went all the way up and came all the way down. So you can see it's foraging really close to the shelf break, on and off the shelf break probably. And then it starts to come down. This is November, and that's December. And it goes off there. So again, a complete cycle. Fishes down off of Florida, goes all the way up to the shelf break up off of eastern Nova Scotia, and then comes all the way back down into the Caribbean.
It's crazy how wide it went when it was coming back down, comparative to its journey out, how wide it was.
Yeah, sort of went past Bermuda on the way down. Yep.
It makes sense why you're getting so many off the Keys. They're cruising through there.
Yep, tagged off the Keys, and then this one didn't. Well, as far as the tag duration goes, this one didn't go back to the Keys in Florida, but it may very well have if the tag stayed on long enough. But this tag stayed on for quite a while.
Oh, that's cool.
That's so cool, man. Tagging data is so fun.
Yep. This is just a profile that Eric Orbison from NOAA provided to me. So this is can you see this or no before I go any further? Okay.
Yeah, vertical profile.
Okay, yeah. So this is a vertical profile of one of those swordfish. And sorry, we do everything in metric, so apologies. But the depth is in meters. If you want to just round it, multiply these numbers by 3. So 600 is around 1,800 feet down. 800 is 2,400 feet down. And you can see that these fish have very regular patterns. Where we have the temperature data matched to the depth data, it's color coded as a heat signature. So the more red it is, the warmer it is, and the bluer it is, the cooler it is, if that makes sense.
Yeah.
And your greens and oranges will be kind of in between.
In betweens.
Yeah, in between. So again, this isn't a really long section of time, but it's just to illustrate how much vertical movement these animals are undertaking. And when we do some calculations to try to get roundabout numbers in terms of how far these fish are traveling. But what the tag isn't really able to do is to capture all of the individual angular movement, the ups, the downs, the lefts, the rights. It only has so much data and so much battery, so it can't physically do that. But when you look at something like this, you got to imagine that, these fish are moving tens of thousands of miles a year, easily, without question, if you count all of those movements.
Not even.
And, this fish is, with regularity, this fish is going below 1,200 feet consistently. There's a couple of times, maybe right here in the middle of the profile, where it's a little bit shallower, but before and after, and then again, it's going down beyond 2,400 feet.
Wow, that is impressive.
Yeah. And swordfish, they have one of the largest horizontal ranges of all the highly migratories, and they can swim very, very deep, very, very deep.
So it's, I mean, that's non-distant.
Oh, that's cool. I always like looking at this sort of stuff and just trying to see
So
what.
I don't know if you can see these. This is just a quick snapshot of yellowfin data. So same thing, color coded, and just a few days. We just took a few days to illustrate what the profiles look like. I mean, interestingly, again, this is Atlantic. This isn't Pacific or Indian, but where we are off the east coast of the U.S., yellowfins can go down to 50-degree water, 45, 50-degree water, 55-degree water. And, for this one particular fish anyway, it seems to do it with quite a bit of regularity. You can see a lot of blues in here where this fish was going down to 1,000 feet.
Oh.
Normally, I think a lot of us, me included, we think of yellowfins as a strictly epipelagic fish, which they are. I mean, if you looked at time spent at depth, almost all of their time would be spent in the upper 200 meters, but you can't rule out that they don't go below it at times.
Yeah, well,
that's awesome. I mean, that was going down pretty quick, like almost regularly in that particular job.
Yep.
It's chilling down there.
Yeah.
So
cool.
Yeah, so all kinds of cool tagging. Tagging stuff done by a lot of different folks around the Atlantic.
Thanks for sharing
Pacific
that.
and Indian Ocean.
Yeah. Yep. No, that is very, very interesting stuff with those. What I wanted to kind of shift on and talk about too, we haven't discussed it, but and correct me if I'm pronouncing it wrong, but this is it Skilly
Project. Yes,
Skilly
exactly.
Project. Yeah.
Yeah,
So I really wanted to get into that before we run out of too much time here and go into
happy
that. And so let's just fire that up.
Sure. So I'll package this or I'll unpack this a couple of different ways. First of all is the umbrella that's overarching for billfishes in general, and the fact that we don't have a lot of information on that. Second to that is within that box, we don't know a whole lot about their migratory behavior. And what we do know about their migratory behavior for billfish often focuses on blue marlins, more so than any other of the billfishes. And so the Skilly Project was born out of the idea that we need more information on the vertical and horizontal movements of white marlin in the Atlantic. There's a ton of outstanding questions. One of which is, do they have this fidelity to certain regions? Do they return to those regions year after year? Where are they spawning? Where do they go, presumably, to spawn? Do they cross from one side of the Atlantic to the other side of the Atlantic? Are there size-specific migratory patterns? And the Skilly Project is an initiative led by the Billfish Foundation, so Ellen and Peter's group. And the idea behind this is to tag and track what they call these skillies, if I'm getting the name correct, which are these large white marlins that are prevalent off the east coast of the United States for a very short period of time, and they sight fish for them, right? So you go out on very calm days, you're looking for these fish, and you can actually cast almost to them, and you can bring them in on light gear. These tend to be larger white marlins. Actually, Sammi was the one that did the tagging on the project this past summer for me, but I believe most of them tend to be on the larger side. And so one of the ambitions of this project is to get a little bit more information about, where do these fish go? We have 3 major tournaments on the east coast of the United States that focus on white marlin. White marlin is such a huge economic driver. Everything from fuel to supplies, boat building, food. I mean, you name it. It is huge. Mm-hmm. These I'm not trying to single out any tournament. They're all good, but I'll just name those big 3, right? The Big Rock, the Mid-Atlantic, and the White Marlin. Yeah. I mean, they're huge. They're huge events. And yet we know so little about these fish. Are they the same fish? Are we fishing on the same fish, or are they fishing on different fish? And so we had one of our tags come off a little bit earlier than we had hoped. But that fish went from Nantucket, and in Sammy, correct me if I'm wrong, if you remember, I want to say 3 months, it was already off the coast of Georgia/Florida. It was in that ballpark, in I think in that region. Another interesting thing on white marlin migration, some of our colleagues at NOAA recently tagged some white marlins off of Portugal, and they ended up in the Caribbean.
Oh, wow.
Yeah. So there's hardly anything known about these fish. And because they're so popular and they're pursued and they're the end goal of a lot of these tournaments, we're trying to figure out more information about them. And so we've done diet studies on white marlin so far. We are doing the aging work that I mentioned before on them. And so this is the next cog in the wheel. And that's trying to understand movement and migration. Where are they going at different times of the year? And the Skilly project is very specific because it's targeting a specific group of fish that are coming inshore off of New England. So these fish are actually much further inshore. They're not out actually at the canyons. They're up on the shelf quite a bit. And they're only there, for, like I said, for a very limited amount of time in the summer, and then they're gone. And they tend to be bigger, on average. And so we put out a few tags last summer in 2024. Last summer, Peter at the Billfish Foundation was working really hard to get this all organized, and we were working on our end trying to get the tags, and we sort of caught the very tail end of the season. But since then, Peter's been putting a ton of effort. We have a bunch of folks that are on board with this project. Yeti, I think Yeti is on board, and Costa is on board, and Viking Yachts. And so there's this big group of folks who are supporting this initiative, and we're just really excited about it. We have a lot more tags to deploy in 2025. So we're ready to go. I want to say we have like another 12 or 14 tags to go this summer at least already. So we're gonna hopefully catch it right at the beginning of the season this year.
How short term are the tags, Mr. Walt? How short term are the tags, Mr. Walt? You said they're short term. Like, are we talking a couple months, 7 months?
So Peter and I, the strategy that Peter and I adopted was a sort of a hierarchical one. We have some tags that are 6-month tags. We have some tags that are 10-month tags. And then we have some tags that are 12-month tags. And the reason behind that is we wanted to because in the past there have sometimes been some issues with them, coming off at different times unexpectedly that we didn't want to, and also the specific questions that we had and the way that you program the tag, we sort of structured them so that they would pop off at different time periods. I'll give you an example. So the tags collect light levels, which is used as a proxy for location. They record pressure, which is a proxy for depth, and they record temperature. Technically, the tag records that every 10 seconds. So it's recording temperature and depth every 10 seconds, or even maybe more frequently than that. And light levels are just for once a day. The problem is that when that tag pops off, you have a limited battery life. So you can't transmit all of that data. You have to pick and choose what you want. And so if you stagger the tags and you say, okay, I want some tags that are going to do 6 months, some that are going to do 10, and some that are going to do 12, what you can do is for the 6-month tags, you can have it report more of the high resolution data. The 10-month tags, a little bit less, but you get more of a track. And the 12-month tags, a little bit less of that high resolution data, but now hopefully you have that circuitous route.
Track, and you're just piecing all the sides together.
Yes, exactly. It's about balancing, right? Because you're trying to put a little piece of electronics on a fish that never stops moving and goes to pretty amazing depths. And then the expectation is you want that piece of electronic that has been bathed in salt water for 10 or 12 months to come to the surface and report to a satellite and send all its data to you, which they do. But you have to balance what it is that you're asking for a question against the data that you need to answer that question. And so by staggering them at 6, 10, and 12, you can answer the different questions based on the different amount of data that you get from each of those tags.
Yeah, I got you. That makes a lot of sense.
If that makes sense. Yep.
Oh, yeah, for sure.
And again, this is another industry-driven initiative, right? This was totally born out of the industry. It was a couple of folks, a couple of captains that we're working with, Rob and John in Nantucket, that kind of spun up this idea with Peter. And they said, hey, you know, we'd like to learn more about these fish. This is a fishery that's very historic. You know, it goes back 60 or more years off of Nantucket that people have been targeting these fish, and they're sort of an enigma. You know, people just don't know a whole lot about them. And so, again, another one of these industry-led initiatives where they pose the question, we refined it a little bit, and then we bring the expertise on the tagging end and the analysis end. You know, I have a member in the lab who does all the fancy modeling on the on the tag analysis, which I don't do, and can create all of these maps and things that, you know, you all were looking at. And then the end goal, as is the case with all of our stuff, is we will transfer that data once it's complete to ICAT, and they can use it in a management frame.
Right.
That's the idea. Vertical profiles. The other thing too that you can get from vertical profiles is, you know, if you do have interactions with billfish and commercial gears, you can use vertical profiles sometimes of animals as a way to discourage that bycatch. Maybe you can set your hooks deeper or shallower, etc., you know, depending on the vertical profile. So again, it gives you so much more information than just, you know, where did the fish go and how deep did it go. There's an applied side to it. There's something that you can use that data for in fisheries management context to protect the resource.
And this is fairly new, like you guys just started in 2024?
2024, yes. Started in 2024. Hope have a great year in 2025. Knock on wood, Mother Nature cooperates, because she's the one that sort of is the arbiter of your success.
Yeah.
But yeah, the initiative is for 2025, and I think if we can get the support, I think Peter would be in line with us that we would certainly like to extend it to another year. And I think now there's also initiatives to do more of this work in the Mid-Atlantic. So to bring that tagging effort down into New York, New Jersey, Maryland, Delaware, that area, North Carolina, bring it into that area.
That's cool. Well, I look forward to hearing all about it.
Yeah.
Keep an eye on all that. And, yeah, that's interesting. Well, yeah, we kind of touched on a bunch of stuff there. I know there was, I have all these sticky notes where I wrote down all the different stuff that we talk on. We're already at, you know, an hour and 40, and I'm like, oh man, there's a lot. So, you know, I won't hold you guys much longer, but you know, is there anything else you want to you think we should talk about, or anything that we kind of missed that?
I think we did hit a lot of those things. You know, we touched on tagging, we touched on the close kin project, and I touched on the diet work that we do and others do, and how important the tournaments are, you know, to those efforts. So I think we touched on a lot of those things. What I would just encourage is for folks that are interested in these different projects, you know, to reach out. And we are doing everything we can. Like, for example, in this Bluefin initiative, we are doing everything in our power to make it as minimalist as possible. And by that, I mean, I've been on enough boats. I know what happens when, this is not necessarily for billfish, but speaking for tuna fish, you know, I know what happens when 8 of the rods go off at once, right? And everybody's trying to go under and over and over and under and go to this side of the boat and that side of the boat, and it's chaos. And the fish are coming in. You're trying to release them. You're trying not to get hooked. But we do everything that we can with our sampling with the industry to minimize that. So again, we try to ship things and have it as like a package deal. It's really quick. Billfish is a little bit easier. Not so much handling the fish because they can be rather wild, but at least you don't usually get 8 blue marlin on at once. However, when you get one on, they go crazy. But at least I didn't ever get 8 on, but they go crazy. So there's a whole nother logistical challenge. But I would just encourage folks to, you know, participate in these collaborative efforts as much as possible. And again, because these are going to be throughout the world, they're not just us. We're a small, we're only one part of the world's oceans, and the Pacific has tons of tournaments and is a big ocean. The Indian Ocean, same thing. And again, as I emphasize to folks, every fish tells a story. So no matter if you're tagging that fish or you're taking a piece of genetic material from that fish, every single one of them is important. You never know what your fish is going to tell us. Maybe it doesn't tell us anything this year. Maybe 15 years from now, someone recaptures that exact same fish 10,000 miles away.
Yeah.
Those are the things that make it, to me, fascinating. And the other thing that we strive to do is for folks to keep in touch with us. I think one of the things that we're focusing on now, I have another staff member in my lab who's kind of been spearheading this effort, and Sammi has as well, is to really create a dynamic between the science labs and the industry to see this work kind of in real time. What's going on? Okay, we put out all these tags. What did they do? We gave you all these genetic samples. What did they do? Now, granted, scientists are slow, not mentally, but they're slow in terms of our ability to keep up with the pace, maybe. So you might give us a sample and then you don't hear from us for a year. That's not because we're trying to ignore you, but it takes a while sometimes to process stuff. But I do want to say that, I strongly put the emphasis on interaction between the industry and the science labs, and for the science labs to really share your information, because that's, for me, that's the most rewarding part, is to give it back. So, and I don't want to take away from Sammi, because I want, she also was leading the tagging effort for the Skilly project this year. So I defer to her if she has some thoughts on that as well.
Oh yeah, I mean, just on the closed can stuff, I was gonna say, similar to what you were saying about the timeline. I feel like it's really cool to have been a part of that pilot project or project and get collect all these samples, get people interested, and now we've had lots of people that are reaching out and trying to get results, and now we're finally in a spot where we are starting to actually have some stuff. So it's cool to see that come full circle, and hopefully be able to share that eventually, and also to see that it's working, because there's nothing better than that, having proposing a project and having all this effort go into it and then having it come back and be successful. Just it makes it that much sweeter to share with other researchers and also the industry. But yeah, the Skilly project is a super awesome project to be on. Work with some really cool people, and I mean, for me, it's a totally different kind of fishing that I've been exposed to working in the bluefin industry. So it's really cool to see them working their craft and be able to just like spot these. I don't know how they stand up there all day and like spot those fish. It's crazy to me. I mean, their eyeballs must be burning out of their head, but super, super cool to be a part of and be able to work with different parts of the industry, and be able to get tags out. And it's always great to get the perspective of people in the industry and just kind of ground the science, and always make sure that we're going in the right direction with it, and have the industry's interests in the forefront of our minds as well when we're doing the research.
And I'd also add too that these are global fisheries, right? These are global fisheries. But I also would say that, again, maybe this is just my perspective, but U.S. fishermen are in a really unique position. We have the ability to get on the water and access these fish in so many different places, and we have the resources to be able to dedicate to this science. And I think it's imperative that we lead the way. We lead the way. Our industries lead the way, because if not, no data is bad data, in my opinion. If you just keep kicking the can down the road and you're like, well, nobody else is doing it, so why should we do it? That can come back to bite you. I'm of the mindset that let's do it. Let's be the pioneers. Let's lead. Let's be the leaders in this, and then share this, with the scientific and international communities. Because if we don't do it, my fear is it won't get done. I'm a big proponent of us really leading the way. I think that we're uniquely positioned to do that, and it will, in the end, it'll pay dividends, huge dividends for us, I think.
That's great.
Couldn't agree more.
Yeah.
Well said.
Well,
But I did want to say thank you to
Yeah.
both of you for hosting us and having us on.
Yeah.
thank you guys. This is really cool. It was. I learned a bunch of stuff today. So, yeah, that's always good. So I appreciate it. I know it's later in the night there, so and you guys able to do that on a Monday and jump on and talk with.
Not a problem.
It's great.
Yeah. Happy to.
So yeah,
Not a
thanks.
problem.
Sorry, go
add
ahead.
one more no, I just add two. So when he was talking about the Closekin project, we have a form. Like, yes, you can reach out to Walt, but also something that might be easier too, and I can share this with you, Chris, is I can send you the form that is just like direct fill out, and then it goes to one of us, and we can get you kits as they roll in. It's super easy. It just asks like your name, your how many sample or how many vials you want, and then we can get people kits right away. So maybe if you have like show notes or something, you can put it in there.
Yeah,
Easy
email
access
that.
for people.
Perfect. Yeah, if you can email it to me,
Great.
and then, yeah, we'll throw it on there, and then also I'll put up a post as well about
Awesome.
what you guys are doing and see who's interested. I'm sure people would be interested. I mean, I know if it ever goes down to Pacific Blue Marlin and you guys are doing it, there's people here that'll be interested. I'll do it for sure. So,
Awesome.
I know guys that are roofing tuna fishing over there or whatever, they're going to, I for sure think that they would want to do it. It's a really interesting and unique thing. So, yeah, that's awesome. So yeah, I will get the info out, and then I would say that there's probably just a lot of people that either are not aware that that's even going on or that that's happening, or they just don't know how to,
Yeah.
get involved.
I think you're right, Chris. 100% on both of those. I would say.
I mean, yeah. So just getting the word out, and so we'll do the same. And Nick, Nick's down there in the Bahamas. A lot of the boats around him right now fish all over.
Yeah, a lot of those guys are going up and they're fishing. Like, I know most of the guys that are next to me right now are doing Big Rock, Mid-Atlantic. Like, they're just doing the whole East Coast circuit. So, I mean, give me stuff. You need to send them. Some of these guys are some of the better boats. A couple of them are even doing Bermuda too.
I'll email you. I'll put you in the emails, Nick, and then you can get all the info as well. And then, just email me like kind of all the info, whatever, and then I'll send some to Nick, and I'll get some, and then we'll put it out there, and then also everyone that's watched this, they'll be able to go and click on it and check it out.
Great. So, awesome. Well, no better way to get it out. So thank you guys so much for having us.
And I'd end with saying too, if anybody who watches this has questions, we are more than happy to answer questions. So send an email our way or send it our way in some form of communication, and we are more than happy to try to answer. I can't say that I have all the answers, but I'll do my best. We always encourage folks to reach out because it's great to have the knowledge on these things, and we're more than happy to share.
Well, thank you guys. Appreciate it so much. I don't have any more questions. Nick, is there anything you wanted to ask or anything before we head off?
No, I mean, good. Say goodbyes and sign off. But yeah, I mean, I think we had a lot of great. I mean, it's nice to get the thinking cap back on for a while.
Yeah. Now back to fishing.
Yeah.
Yeah. Right on, man. Well, you guys, I'm gonna hit the unrecord button here.